In our previous lesson, we divided the immune response into two parts. The innate immune response, which is non-specific, which does not depend on the identity of the intruder, and we said that its effectors are the natural barriers and the inflammatory reaction or the phagocytes. Today we'll be talking about the acquired immune response, the specific immune response, which is dependent on the identity of the intruder. So how can we prove that the immune response is specific? In this experiment here, we injected the chicken with killed salmonella, killed to attenuate its effect, to make it not virulent, not to kill the chicken. And then 10 days later, we injected this same chicken with live salmonella. The chicken survives. However, when the chicken was injected with killed salmonella, but then with live V. cholera, the chicken dies. This is the similar case where we inject the chicken with killed salmonella and killed V. cholera and then live Vibrio cholera, it survives. But when we injected it with killed V. cholera but live salmonella, it dies. What does this mean? This means that the chicken acquires the immunity against salmonella. It is now immunized against salmonella. So if we inject it with this virulent microbe or antigen, the chicken will survive because it has the ability to fight this antigen. However, when the chicken acquires the immunity against salmonella, but I inject it with live V. cholera, the chicken dies. So this means that the immune response is specific and it is only directed against salmonella because the chicken was immunized only against salmonella. What are the types of this specific immune response? In another experiment, we injected a guinea pig with the tetanus toxoid. The toxoid is an attenuated toxin. It is a weakened toxin to provide or to immunize this animal against tetanus. After a certain time, we take out the blood and we divide it into serum and into cells. Now, the injection of the immune cells along with the tetanus toxin, which is the virulent toxin, causes the death of the guinea pig. So this means that the cells could not help this, even the immune cells, the T lymphocytes and the B lymphocytes, they could not help this mouse to survive. However, if I take out the serum and inject it into a mouse A2 along with the tetanus toxin, the mouse will survive. So this here is a technique called serotherapy. I remove the serum or I take out the serum of a previously immunized animal and I inject them into an animal alongside with the tetanus toxin, it survives. So this means that the serum has assisted or helped this mouse to survive. This means that the serum contained molecules that can help this guinea pig to fight against tetanus. These molecules are called antibodies. And to make sure that these molecules are the antibodies, I filter this serum through a matrix that contains the tetanus toxoid attached to it. And so the anti-tetanus antibodies will bind to this matrix. And then when I inject the filtrate into this guinea pig along with the tetanus toxin, it will die. Because this now filtrate does not contain the anti-tetanus antibodies, which are specific to fight against the tetanus toxin. So in this case, we now know the first effector of the specific immune response, which is the serum, more specifically the antibodies that are found in the serum. In another case, we injected this guinea pig with CGB. CGB or Calmette and Guerin bacillus. It is an attenuated Koch bacillus. KB causes tuberculosis, in Arabic sil. So this is the CGB, which is the attenuated KB. By the way, here they are suggesting now that individuals who are vaccinated against CGB are fighting well against or managing to fight well against the coronavirus because they are both two respiratory antigens or pathogens that attack the respiratory system. Anyway, now this CGB, whenever it is injected into this guinea pig, and later on I extract the blood, I take out the serum and inject it into another guinea pig along with the virulent KB, with the deadly KB, this guinea pig dies. So in this case, the serum did not do any help. However, if I inject the immune cells into a mouse B2 along with the KB, this guinea pig survives. Now, I separate these cells into B cells and into T cells. B lymphocytes or T lymphocytes. The injection of B lymphocytes alongside with the KB to a fourth guinea pig causes its death. However, the injection of T cells causes its survival now along with KB causes its survival. So this means that the second type of immune response is mediated by T lymphocytes. So now I have, depending on the nature of the antigen, if it is, for example, the tetanus toxin or the KB, I can differentiate between two types of immune responses. They are called humoral or the specific humoral immune response and the specific cell-mediated immune response. The humoral immune response is mediated by antibodies in the serum. However, the cell-mediated immune response is mediated by T cytotoxic cells, and later on we will be talking or explaining how do antibodies contribute to the elimination of the antigen and how do T cells contribute to the elimination of the antigen. Thank you very much.